So what we have learned is that if two genes are completely linked, then that means they are on the same chromosome. So remember there are 4 combinations possible because phenotypic ratio was 9:3:3:1. In the F2 generation or in the test cross dihybrid, it will be 1:1:1:1. That means 2 are parental types and 2 are recombinant types. So if two genes are completely linked and are present on the same chromosome, then the ratio should come 50% one parental type, 0% first recombinant type, 0% second recombinant type, and 50% again the second parental type. That means if two genes are completely linked, 0 recombination and 100% parental types, 1:1. That's why this ratio is called as 1:1 ratio. I hope this is clear. Now why it is 1:1 ratio? Because these two genes are not separating. And why they don't separate? Because during crossing over, see, if two genes are present like this, imagine this gene and this gene. So if during crossing over, crossing never cause them to separate from each other, then what will happen? They will go as such during every gametogenesis. That's why these genes are highly linked genes. So when two genes do not separate during crossing over, during meiosis first, then you will call the ratio will come out in the test cross would be 1:1 ratio, or only parental types are formed, recombinant types are not formed. If two genes are unlinked, so two genes can be unlinked also. If they are unlinked, that means they are present on different chromosome. Because they are on the different chromosome, if you remember the test cross ratio of the dihybrid in the Mendel's case, because that capital Y and capital R or small y and small r were present on different chromosomes. So if two genes are present on non-homologous chromosomes, they are called as unlinked genes. In that case, the ratio of the parental type, recombinant 1, recombinant 2, parental type 2 will come out to be 25%, 25%, 25%, 25% each. That means 1:1:1:1. Now remember, this is even possible if the genes are present on the same chromosome, and that is in very rare cases, and that will happen in a case when two genes are present quite far from each other, and that's why whenever crossing over occur, they are going to go separate. So when two genes are very far on the same chromosome, they will go away maximum time, and then there are high chances of 1:1:1:1 ratio formation. If two genes are incompletely linked, that means they are on the same chromosome but not so far. So every time, see, when I say when two genes are unlinked, so the ratio should come this much. If they are present on the same chromosome, the condition is what? Every time there is meiosis, they are supposed to be separated by crossing over. So, but when two genes are incompletely linked, now what is incompletely linked? That they are present on the same chromosome, but every time there is meiosis, they do not separate. Not every time. Sometimes it is possible. So when sometimes it is happening, that's why sometimes only recombinants are formed. That's why chances of recombinants are less and chances of the parental type are more. So if two genes are incompletely linked, that means they are present on the same chromosome, parental type 1, parental type 2 will be more than 50%, or you can say more than 25% each, and the recombinant type will be less than 25% each, or you can say less than 50%. Reason is what? Because not every time they are separating, sometimes they may separate. That's why the ratio which came here is not 1:1:1:1. The ratio is 7:1:1:7. That means out of many, only one time it happened that they separated, and the ratio was 1:7 times. So more time they were not separating, only rarely one time they have separated. So independent assortment is possible in two conditions. What is the first condition? Independent assortment is possible if two genes are on the different chromosome, that means they are on the non-homologous chromosomes. And second reason, independent assortment is possible if two genes are present on the same chromosome, but they are far enough that every time there is crossing over, they are, so please remember, every time. That's why it is not possible every time. That's why the ratio, generally the genes are incompletely linked. Maximum genes on a chromosome, on a single chromosome or homologous chromosome, would be incompletely linked genes because they will separate, but not every time. Now the next is frequency of recombination. So how many times the recombination is taking place, or you can say the crossing over frequency, CO mu. So crossing over frequency, this concept was used by Alfred Stewart Wynne. Remember, he was the student of Morgan. So I told you in the last class, Morgan gave this world brilliant students. So one of his students was Alfred Stewart Wynne. So this person used this frequency of recombination for gene mapping on a chromosome. That means you can easily tell which two genes are close to each other, which two genes are far. So you can measure the distances between genes in a relative manner, not in the measurement, but in the case of relative manner. So he gave these terms. To honor his teacher, he started using, like we say centimeter, he said centimorgan, then decimorgan, then 1 morgan. So 1 morgan is equal to 100% crossover. So when two genes are very far from each other, so that every time there is a crossing over going on, they will always separate. So 1 morgan is equal to what? 100% crossing over. 1 decimorgan is equal to what? 10% crossing over frequency, and 1 centimorgan is 1% crossover frequency. Let's understand a small numerical to understand this concept.